We are now ready to talk about gene expression, the two phases of gene expression in more detail. We're going to start with transcription first because it is the first stage of gene expression. Remember that transcription is the creation of RNA. This process is going to be catalyzed by an enzyme that's called RNA polymerase. Very much like when we are replicating DNA, we use DNA polymerases to copy the DNA or to build new DNA strands. When we want to make RNA in transcription, we're going to use RNA polymerases to accomplish this job. RNA polymerases are different, however, from DNA polymerases. Remember that DNA polymerases were very much divas, and they had a long list of things that they could or could not do based on their circumstances. RNA polymerases, you can think of them more like a one-man band. They can do everything when it comes to creating new RNA strands. So, for example, helicase is not needed. RNA polymerase will pry the two DNA strands apart on its own. You don't need single-strand binding proteins. RNA polymerase stabilizes single-strand DNA by itself. You don't need a primer. RNA polymerase can start from scratch, adding new RNA nucleotides to match the DNA template. So RNA polymerase is a much easier enzyme to deal with. Again, it's more like a one-man band. It kind of does it all itself. When the RNA polymerase builds the messenger RNA, remember that the mRNA is complementary to the DNA template strand. And as I mentioned a minute ago, RNA polymerase does not need a primer, so it can start from scratch. However, it does build the new RNA in a 5' to 3' fashion, just like DNA polymerase. It cannot run backwards. And then when we're thinking about as the RNA polymerase builds the RNA complementary to the DNA template, remember there are the same base pairing rules, right? G's pair with C's, and A's would like to pair with T's in the RNA, but remember there are no thymines in the RNA. Instead, you're going to be using the uracil. All right, so as we start talking in more detail about transcription, transcription takes place in 3 stages. Initiation, how do you start the process? Elongation, how do you make the mRNA longer? And then termination, how do you stop this process? And we're going to go through each of these 3 steps in more detail. The thing we're going to do first is actually look at transcription only in prokaryotes, because again, in prokaryotes, this process is simpler and more easily understandable. And then what we're going to do is we're going to layer on top of that, how do eukaryotes do things a little bit more complex. So this is prokaryotic transcription, the basics: initiation, elongation, and termination. So what we're looking at here is a piece of DNA. And this piece of DNA has a gene on it. And so the gene here is kind of in the dark blue color. And this prokaryotic cell would like to transcribe this particular gene. So first, what happens in transcription initiation in prokaryotes is that the RNA polymerase, which looks like this little guitar pick protein here, it will bind to a region that's upstream of the gene called the promoter. The promoter is the place where the RNA polymerase docks. Once the RNA polymerase finds the promoter, then it can bind to the DNA, pry apart the 2 DNA strands, figure out which one's the template, and at the start point of transcription, it will start adding complementary RNA nucleotides to match the DNA template, much like what you see here. So the DNA is in blue or seafoam color, and then the red are the RNA nucleotides that are being added complementarily to the template to start building this RNA transcript. That's initiation. Elongation. So now that the RNA polymerase has started building this mRNA, it needs to make it longer. And so what the RNA polymerase will do is it will literally just move down the DNA, opening up the DNA that it needs to access in order to be able to continue production of the mRNA, and just sort of chugs along down the gene. And as it does so, the RNA transcript gets longer and longer and longer and sort of hangs out the back of the RNA polymerase. And notice that the RNA is being made in a 5' to 3' fashion. So the new nucleotides are always being added here to the 3' end. So how do you stop this process? Well, for prokaryotes, it's actually pretty simple. The RNA polymerase will run into a sequence at the end of the gene that's called the terminator. It's in the DNA, and the terminator essentially tells the RNA polymerase, you're done, this is the end of the gene. The RNA polymerase is going to hop off the DNA, and then it'll release the completed RNA transcript, which then can be immediately used for translation.